Welcome back, students. Today we'll be discussing about the different types of modifiers and the gene interactions. Previously, we have discussed about the law of independent assortment and the Mendelian law 2, which enables the two different organisms or the species to form 16 different types of progenies into four different phenotypes and three different genotypes. Ultimately, they tend to produce the four different types of phenotypes with the ratio 9 is to 3 is to 3 is to 1. Today, we'll be discussing about the other modifiers which tend to deviate from the original law of independent assortment and restricting the ratio of 9 is to 3 is to 3 is to 1. So, under which we will be discussing about the supplementary gene interactions, complementary gene interactions, and we will also discuss about the epistasis, under which we have dominant epistasis and recessive epistasis. So, we will discuss about all these four different types of gene interactions or the deviations from the law of independent assortment, citing one example each as per the syllabus. So we have already discussed about what is the difference between the character to the of a trait. Usually, a color could be a character under which we have two different traits. It could be white or it could be black. So the different colors forms the different traits, which falls under a single type called a character. Another example would be height. So height could be considered as a character, and the different types of height, like tall and a dwarf, could be the traits. So a single character can be governed by two or more genes. Earlier, we have understood that one gene could form one factor or it could give rise to one trait. But now it's time that we need to understand that also more than one gene at a time could form more than one given trait. And we call them as a non-allelic or intergenic gene interactions. That is, the interactions of more than one gene leads to intergenic gene interactions. The independent genes located on the same or the different chromosomes tend to interact or correlate in order to form a, a single phenotypic trait. So the first one that we will be discussing is about the epistasis. Epistasis, as I told you, is a process or a mechanism which is formed due to intergenic suppression or the masking effect. So, to put it in a simple words, we need to understand that one gene tends to dominate and the other gene which gets repressed or which is masked by the action of a dominant gene is considered as a recessive epistasis. So, as I told you in the beginning, there are two different types of epistasis. One is a dominant epistasis, and the other one is the recessive epistasis. So these two variations or gene interactions tend to give out different types of results. So a gene which tends to express itself in the presence of another gene is considered as a dominant epistatic gene. So the gene or a locus which gets suppressed by a dominant epistatic gene is considered as a hypostatic gene. Generally, the examples are the body color of many tropical fish is controlled by such epistatic interactions. So, the dominant epistasis occurs when a dominant allele at one given locus or a position of a gene tends to produce a particular phenotype. So here phenotype refers to a trait. It could be the hair color, uh, the height, or it could be any given phenotype. regardless of the genotype at the second locus. The second locus here is nothing but it could be a recessive allele. So the second gene can express its phenotype only when the epistatic locus is homozygous recessive. That is, if the homozygous allele, which was dominant earlier, if it is in small t and a small t, or a small a and a small a, only under such circumstances, the second locus can express itself, which we call it as a recessive epistasis. So here, the type of epistasis and the ratio is the common car scale pattern. It is formed due to the dominant epistasis, and the F2 phenotypic ratio would be 12 is to 3 is to 1. And the recessive epistasis, the Mexican cave caracins, the eye color, the trait could result in 9 is to 3 is to 4. We have many examples. We could discuss about the coat color in mice. We could discuss about many such examples in the subsequent videos too. So the recessive epistasis occurs when the recessive alleles of one locus, for example, AA, that is, if it is homozygous recessive. You should know the difference between homozygous dominant, heterozygous, and the homozygous recessive. So this is the homozygous dominant. That is, both are capital letters. This is the heterozygous. That is, it is a mix of both the alleles. And this is again a homozygous recessive alleles. So recessive epistasis occurs or expresses itself when the recessive alleles of one locus, for example, AA or BB alleles, suppress the phenotypic expression of the alleles of another gene. For example, it could be capital B, or heterozygous allele. This type of epistasis is called as recessive epistasis. Another term that we need to understand is the pleiotropism. The phenomenon of multiple effect, that is multiple phenotypic expression of a single gene, is called pleiotropism. So pleiotropism is defined as an expression of a particular trait by the action of multiple genes. So one gene has got its effect on different parts of the different characteristics of one and the same organism. This is how we understand pleiotropism. So in the subsequent videos, we will discuss about the supplementary gene interactions and the complementary gene interactions. Thank you, students.